Good day everyone, this is Johann and welcome to my second cycle RD videos in microbiology. The first thing we should do after donning the PPE is to disinfect the working area. This part is a continuation of our first cycle videos, the reading of results in biochemical tests. Antimicrobial susceptibility testing. First, we should label our plate. We will now make a suspension to compare it to the Macfarlane standard. Macfarlane standard are used as a reference in order to adjust the turbidity of the liquid or the bacterial suspension in the vial or tube in the microbiology laboratory. If the inoculum does not contain a concentration of bacteria that approximates the 0.5 Macfarlane turbidity standard, antimicrobial susceptibility test results will be affected. This helps to maintain and ensure that the number of bacteria will be within a given range to standardize microbial testing. For instance, a resistant organism could appear susceptible if too few bacteria are used in the inoculum. The density of the suspension of bacterial cells is compared to the Macfarlane turbidity standard by holding the suspension and Macfarlane turbidity standard in front of light against a white background with contrasting black lines. To Kirby-Bauer disk diffusion susceptibility test determines the sensitivity or resistance of pathogenic bacteria to various antimicrobial compounds in order to assist physicians in selecting treatment options to their patient. Lawn cultures are prepared by evenly spreading a high amount of bacteria onto an agar plate using a sterile cotton swab soaked in the bacterial culture or suspension. After the first swab inoculation with organism, the second swab inoculation is rotated 60 degrees or 1/3 to first inoculation. Use same swab and don't re-inoculate it. Remember to carefully cover all the surface area without leaving gaps or spaces. For the third swab inoculation, rotate the plate and as well as use the other side of your swab, but don't re-inoculate the swab. Then, allow the plate to stand for 10 minutes in room temp. Once the lawn of bacteria has been created, the antibiotic disc can be applied to the plate. Antibiotic disc should be removed from fridge or freezer before use and allowed to come to room temperature before use. The disc can be applied using an antibiotic disc dispensing machine if you have one in your laboratory, or use sterile forceps if you do not have a dispenser. When using the forceps, you should take care not to rip or damage the disc when handling it. Ensure that you press the disc down onto the agar so that it is secure and does not fall off. If using forceps, this can be sterilized between uses by dipping the tip of the forceps into ethanol and passing through a blue flame. When undertaking this diffusion testing, it is important to get the antibiotic disc onto the plates within 15 minutes of creating the bacterial lawn. Once done, invert the plate for incubation. Incubate the plates for 16 to 20 hours at 35 degrees Celsius. After incubation, now it's time to read and interpret the zone of inhibition. To determine whether the bacteria are sensitive to the antibiotic disc you have placed on the agar, you will need to measure the zone of inhibition around the disc from the back of the plate. You can measure the zones using a ruler. The measurement should be the diameter of zone of inhibition where there are no bacteria have grown to the nearest millimeter. AFB smear preparation and staining. Prepare a clean glass slide and label specimen number at the frosted end of the slide. To make a sputum smear, fish out 1 loopful of yellowish particle of sputum and spread evenly on slide with an applicator stick. Apply the sputum following the template. Cover the whole area before coiling. When you start coiling, always remember that the smaller the coils, the better. After doing so, allow the smear to air dry completely at room temperature. Place the slides on a staining bridge and pour carbofuscin on the whole surface of the slide. Heat the slide till steam comes off from the stain. Do not boil and do not allow the slides to dry. Leave it for 5 minutes. Remove the excess stain by tilting the slides and wash with water. Decolorize with acid alcohol. Then, wash with water and tilt the slide to drain the excess water. Counterstain with methylene blue for 10 seconds and remove the excess stain by tilting the slide and wash with water. After doing so, place the slide on the slide rack to dry in the air. After drying, place a drop of immersion oil and examine the slide under OIO. Identify the presence or absence of acid-fast bacilli. Lastly, report the result according to the National Standard Reporting Scale. Burn the used applicator stick before discarding.